The reduced coenzymes that are formed in the citric acid cycle are high energy molecules, and they utilize their energy in order to produce additional ATP. In the electron transport chain, the energy from these reduced coenzymes is released in a series of redox reactions that move electrons from one electron carrier to the next. As each carrier is reduced, meaning it gains electrons from the preceding carrier, and then is oxidized, which means it loses electrons by passing to the next carrier. Each reaction is favorable, meaning energy is released, so they're exergonic. And the electron transport chain is the series of biochemical reactions that pass electrons from reduced coenzymes that were formed in the citric acid cycle to oxygen and is coupled to ATP formation. So let's take a closer look at this. And so in the electron transport chain, I'll show you this in a second, each reaction is going to release energy until hydrogen ions react with oxygen in order to form water. The electron transfer will also cause hydrogen ions to be pumped across the inner mitochondrial cell membrane, and this creates what we call an energy reserve, and this is what's used to synthesize the ATP. There are 4 enzyme complexes that have all of the functional groups that are needed in order for these redox reactions to occur. There are various cytochromes. These are proteins that contain heme groups, and iron will cycle between iron 2 and iron 3 ion. Electrons are passed from the weaker to increasingly stronger oxidizing agents, and there is a release of energy for each transfer. So remember from before I showed you a heme group, so this is a heme group. And here is a representation of a cytochrome. And we see this green ribbon here. This is an amino acid chain. The heme group, of course, is in red. And the iron atom is here in gray. Coenzyme Q, also called ubiquinone, because its ring structure with the 2 ketone groups right here, so 1 and 2, is a quinone. Okay, so let's take a look at what's going on here. So here we have hydrogen ions that are being released for transport through the inner membrane at complexes 1, 3, and 4. And some of these hydrogen ions also come from the reduced coenzymes. Remember, they have the hydrogens. So we have the NADH and the FADH. Notice the NADH that interacts with complex 1, and here we have our FADH. Okay, so here is the cytosol. This is the outer mitochondrial membrane. This is the intermembrane space with these hydrogen ions. Here we have the inner mitochondrial membrane and the mitochondrial matrix right here. Also notice the hydrogen ions. Notice hydrogen ions, okay, are able to pass into the intermembrane space, okay, through complex 1, 3, and 4, okay. And then notice here that the hydrogen ions, okay, are able to enter the mitochondrial matrix through this ATP synthase. Okay, so this is like a hydrogen ion channel that allows the hydrogen ions to pass from the inner membrane space into the mitochondrial matrix. And here we see the ADP, okay, and that is phosphorylated, producing ATP. in the mitochondrial matrix. So some of the main points here. The hydrogen ion concentration difference creates what we call a potential energy difference across the two sides of the inner membrane. It's very important that this concentration gradient across the membrane be maintained, because this is the mechanism by which energy for the ATP formation is made available. So the energy release drives the phosphorylation of ADP, right? So the conversion of ADP to ATP. And ATP synthase, that's the enzyme complex that I just showed you in that representation. This is the enzyme complex in the inner mitochondrial membrane. And this is where, as I said before, the hydrogen ions cross the membrane, and which allows ATP to be synthesized from the ADP. And then finally, we have oxidative phosphorylation. And this is the synthesis of ATP from the ADP. The energy to synthesize the ATP is the energy that was released in the electron transport chain. The reaction is both an oxidation and a phosphorylation. So the energy that is released from the oxidation of the reduced coenzymes, so the hydrogen ions return to the matrix and are used to convert the ADP to ATP. And of course, that takes energy. And that energy comes from the oxidation of those reduced coenzymes. ATP yield from oxidative phosphorylation. Well, recent research suggests that each NADH molecule will yield about 2.5 molecules of ATP, and each FADH2 molecule yields approximately 1.5 molecules of ATP. Okay, this is just another slide. It shows how the hydrogen ions return, we see that right here, return to the matrix by passing through this channel that is part of the ATP synthase enzyme complex. And so this is the conversion of ADP to ATP. So again, both an oxidation and a phosphorylation. So I got a quick question for you. Is the pH higher in the inner membrane space or in the mitochondrial matrix? This is the mitochondrial matrix. This is the intermembrane space. So where is the pH higher? Well, we see a lot more hydrogen ions in the inner membrane space, okay? And we see fewer hydrogen ions in the mitochondrial matrix. So the fewer hydrogen ions means what? It means a higher pH. So pH is higher in the mitochondrial matrix. Just a little test of acid-base chemistry.